Hello, everybody. Welcome to the Grit City Think and Drink. I understand it's a Tuesday, really kind of a Monday, but still. And I understand that it got dark 4 hours ago, but thank you all for waking up and coming out tonight. Come on in wherever you can find some free space is fair game. I can always dig out some more chairs if we need them. My name's Jim Gall. I'm a professor at UW Tacoma and the host of the Grit City Think and Drink. This is the end of almost the end of our 7th year of doing this. So we've been going for a while now. I'm super excited about that, but we have speakers lined up for 2025 as well. We did have our speaker for December 10th drop out today. So if you have any ideas, we're trying to shuffle things around. Pay attention to the website. We'll send out announcements. If you want to get on the Grit City Think and Drink listserv, you can scan that QR code and actually add yourself. I do not send out anything besides announcements and calls for personal money. So as long as you guys are okay with that, go ahead and add yourself on there. But you can always go to the Grit City Think and Drink website and check out past talks. We record them. Sometimes I do it right, and you can actually hear everything they say. Sometimes I do it wrong, and you can see the person miming in front of the audience, and you can see their slides. So hopefully you can go check those out and see what's coming up and what you've missed in the past. But that's the scoop. So for how many people are new to the Grit City Think and Drink that are here tonight? Some newbies? Yeah, all right. We hope to have you back for sure. So again, check those out. But the way this runs is I will introduce our wonderful speaker. She'll give you a talk to knock your socks off. Then she'll take a short break. We will give away a couple free swag items. So hold on to your raffle ticket or we'll get you one. And then you can play Stump the Speaker after that with your questions. And that'll do it. Sound good? All right, here we go. So without further ado, the guy who can't read, come on, is going to introduce your speaker. Leighann Chaffee is an associate teaching professor. We'll take the associate off soon, hopefully, at the University of Washington, Tacoma in the School of Interdisciplinary Arts and Sciences. Her research and teaching intersect at her training in, I'm totally botching this. Her training's in neuroscience and interest in health behaviors. Leighann has published pedagogical materials for neuroscience and the psychology of eating, including her 2022 textbook, A Guide to the Psychology of Eating, which we will do a reading group and self-diagnose on afterwards. She is also dedicated to high-impact practices for students from study abroad, from research to global learning, and teaches a study abroad course in food and culture in Prague. Yeah, I gotta go to that. Outside of UWT, Leighann enjoys beachcombing with her family, who's here tonight. Elaborate baking projects, which I was just asking about, and occasional running. All right, without further ado, let's welcome our speaker.
Okay, how's my volume? Is that all right? All right, thank you so much for being here tonight. Let's get some slides. Awesome. So my goal is to highlight some awesome work that was already completed by students at UW Tacoma. And the way that it highlights some of the limitations of traditional approaches used to study eating. Additionally, I'm gonna move this over a bit. I scheduled this talk to motivate me to start working on my next project, so I'll introduce that at the end. So I am in my 14th year at UW Tacoma, and in grad school, I studied behavioral neuroscience. And what that looked like was if you give a mouse, or in my case, a rat, a cookie, it eats it enthusiastically, even when it's already full, which is not unlike myself. I'm generally interested in studying motivated behaviors like eating, or drinking beer, or spending time with people that you love. And when I came to UW Tacoma towards the goal of developing a research program that was really inclusive of undergraduate students, I now study health-related decision-making. So we ask questions like, how does stress and its impact on cognitive load influence food preferences? So still a biopsychological lens. And so I want to highlight some work that's done by students who have graduated from UW-Tacoma. Lexi Luna graduated last year, and she's probably getting ready to interview for medical school. And she was curious if a brief intervention that uses metacognition could impact food choice in an acute setting. And so she did an online study where the participants could fill up their shopping basket with beverages, mostly sugar sweetened beverages. We didn't have alcohol as an option, though that would have been really interesting. And then she had some specific prompts to guide metacognition. So metacognition means our thinking about our thinking, the knowledge that we have about our thought processes, the way that we monitor, control, and evaluate our cognition. And then in the same setting, they repeated their beverage selection. They could adjust what was in their shopping basket. And they answered some questions about psychological variables like stress and some demographics. So these metacognitive prompts look like, how would you describe your thought processes when you select a beverage to drink in your day-to-day life? Or what types of things do you consider when you make the choice of what to drink? And this was effective in the short-term setting. The average participant reduced their sugar grams in their shopping cart by 18. So it was like an entire sugar sweetened beverage, right? So this is cool, but it's pretty obvious what we're asking participants to do here. So there's a chance they were just behaving as they thought that we wanted them to. There's a response bias. And also this study replicated some of the other problems that we found in like 10 years of this health-related decision-making research. So what are these problems? So the scholarship about food choice and decision-making towards our diet is pretty vast. But in our lab, where the participants completing the work are UW-Tacoma students, we fail to find some of the expected relationships between key variables and health outcomes. So for example, some individual factors, one of the factors that's studied quite a bit is dietary restraint. It's the extent to which someone is watching what they eat, right? Or dietary disinhibition, the extent to which someone just overeats more than they anticipated. And then more obvious factors like stress and cognitive load. We failed to find some of the relationships between health choices and health outcomes, like whether people consume sugar sweetened beverages. And we studied that by having folks have access to them in the lab or just having them self-report their consumption, their food preferences in general, or demographic indicators. These are generally indicators of health disparity that are pretty robustly related to health outcomes like socioeconomic factors, gender, and ethnicity. Okay? So answering the question of why our findings were unique is really something that I want to credit the students I work with when their like fresh perspectives and their critical lens towards this, I think really enrich the research program. Sorry, 1 second here. So there's this large and established scientific community that studies food choice, and really the outcome they focus on is weight, which I think is problematic, right? And when they think about this, they use body composition as a proxy for health, and specifically they use BMI. BMI is a metric of body weight per body height, right? And so we see that in decades of research, like 4 decades of research, this is the dominant outcome in health-related like food decision-making work. And this matters because BMI is not a reliable predictor of health outcomes. You may have seen it like in your medical chart. It's especially not a good predictor in adults. It's not a good predictor of adiposity, and it's worst for females and for people who are African American. And I think it's important to note that when it was initially designed, it was to measure population averages. It was never designed to measure individual proportions. So it's weird how much we use it today. And the person that first published on it was actually a follower of eugenics. He was trying to define population averages for young men in Germany in the 1830s. So it's weird to me that we ever adopted it based on this, right? So it's problematic in its origin, and it's not inclusive in its application, and it doesn't really actually measure health. So an alternate that we don't use is a term called overnutrition, which is a form of malnutrition in which excess intake over time impairs health. Sorry, I need to mess with this for 1 second. like I can't like click over here and make this smaller. Oh, there it is. Okay. Okay.
Good.
Thank you. So, in today's world, we see that rather than using overnutrition or even measuring health outcomes directly, certain BMI cutoffs are used to basically medicalize body shape, particularly people who live in larger bodies. And there's some consequences to this that I'm going to talk about in just a few minutes. Okay, another thing that the students I worked with noticed was the universities where the research on food choice was being conducted. It's a long known problem in this research area that research in health, in social sciences, broadly speaking, is not very representative. The work on food choice, some of it is in the US and Canada and Australia, but the majority of it comes from the Netherlands and Belgium. So a pretty specific and narrow population that's not representative. We call this problem the problem of weird participants. So a lot of research in this area comes from people who live in societies that are Western, educated, industrialized, rich, and democratic. If you look at the world's population, only about 12% of people live in these weird societies, but a majority of the research comes from it. Okay, so this raises some questions about how we understand health. This lack of representation is a further limitation. Oh yeah, so Yemi Chawla, who graduated last year as well and is currently in a post-baccalaureate program in neuroscience at University of Pittsburgh, compiled data from several projects over a few years, noting that our sample of participants from our wonderfully diverse campus was probably different than the folks coming from the Netherlands and Belgium that were largely studied in this area. Okay? So she made this really beautiful graph. You see on the y-axis, it's the proportion of individuals who would have a BMI over 30, right? So it's just the general health metric of obesity. There's total on the left, and then men and women. The lighter bars in the background are from a larger NIH data set of several thousand participants. And then the darker bars in the foreground are from UW-Tacoma, several hundred participants. And you can see here that there's some discrepancy between the darker bars in the foreground, the data from UW-Tacoma, and the lighter bars in the background. We're not showing quite the same pattern you would expect. The data is a little bit messy for males because not as many males participate in our research, but if you look at total and females, you can see some things are showing up in a different pattern here. And what we see is there's no significant difference in body composition by ethnicity or income in our UW-Tacoma sample. This is really different than you would typically expect. But even more, you don't see the typical patterns of diet quality that are different by these metrics of health disparity. You don't see the same differences in diet quality across gender or ethnicity or socioeconomic status. And to me, this calls into question some of the patterns that we observed. I mentioned earlier, we failed to find some of the expected relationships between individual factors and BMI. And one reason could be is that those relationships were established with weird samples. Okay. And Yemi says, well, maybe we think folks are unhealthy, but really they're not, right? Is kind of her perspective on it. So these two problems so far are really problems of measurement, problems of the participants that we're sampling and the way that we're operationalizing health outcomes. But there's another problem related to measurement as well. When we study health outcomes, we need to know what people are eating. And that's difficult to measure directly, right? So we rely on memory-based dietary assessment methods. The NHANES, or the National Health and Nutrition Examination Survey, uses these type of instruments. They might ask you, what did you eat yesterday? Or the NHANES actually uses the food frequency questionnaire. So how many tomatoes did you have last week or last month? What about tomatoes cut up in salads or on sandwiches? Tomatoes mixed into sauce, like marinara sauce or pizza sauce? What about in ketchup or salsa? I couldn't accurately report how many tomatoes I had last week or last month. And I think it's pretty obvious there's a problem here, okay? So these memory-based dietary assessment methods that are used by NHANES and other large nutrition surveys in other countries rely on self-report, and they're retrospective, asking you to look back and remember what you consumed in the past. The results from these assessments are used with databases to calculate total energy consumed, as well as macronutrients and micronutrients. And this is iterated into the research and recommendations that we receive as the general public, like the Choose My Plate plan or previously the Food Guide Pyramid. That's where they come from. It's like from NHANES data, in part. And you might not be surprised, this results in significant problems and underreporting. Analysis of 4 decades of the NHANES data showed underreporting so significant it was physiologically implausible to maintain life. And what's more, the underreporting was selective. No one's underreporting how much kale they eat. And it increased over time, even as survey methods improved. Okay. So like, why is this? One, our memory is imperfect. When you go back into your long-term memory, recall information, work with it, and then reconsolidate it, it's vulnerable to revision. It's prone to error. 2, calculating the energy from a database. This is really hard when we're making portion size estimates or trying to understand the composition of a meal that's composed outside of the home, like at a restaurant, like the pizza we had tonight. And there's also this problem of social desirability, right? The problem is of underreporting. No one's reporting that they ate a whole pizza, right? And so the social desirability bias shows up again. And I think this is really important because public health experts and public health organizations say or identify that data collection is the first step in solving a health problem. So if the data collection is flawed, that means that we can't really ever understand that health problem. Oops. Okay. So we've identified 3 possible explanations for the fact that the data I've collected doesn't really make sense, right? I'm trying to rationalize it a bit, but I think there's also reasons. 1 is related to how we operationalize health outcomes. 2 is related to participants in our sample versus generally that are reported in food decision-making research. And 3 is related to measurement, particularly measurement of diet quality. I think this highlights some more general limitations on research on health behaviors. But what we see is there's actually a lack of consensus in parts of the scientific community on the use of BMI in disease classification. And there's something that's demonstrated called a white hat bias. This means that there's a distortion of information in the service of what is perceived to be righteous ends. Publications that call into question the use of BMI are discounted, or worse, there's actually tales of researchers being ostracized in the scientific community for challenging the assumption that the BMI is a useful metric. But I think even more important are the consequences for individuals. With the medicalization of certain shapes, we've seen that there's bias towards people in bigger bodies with the media coverage of the obesity epidemic. This anti-fat bias has increased over time, and up to 80% of Americans endorse anti-fat beliefs today. This causes a cycle of harm from weight stigma. When people who live in bigger bodies typically receive discrimination at work or in a healthcare setting, this increases their stress. They may be more isolated. This undermines their ability to cope with this stress, their ability to emotionally or cognitively self-regulate, which further harms health. So how do we do better, right? My question is, how can we, rather than focusing on these outcomes or consequences of eating that perpetuate the system of oppression in the metrics that are used, I'm actually interested in turning towards more positive emotions like the joy that comes from eating. Here's Rosie having her favorite ice cream. She's here. Unfortunately, I mentioned before that my background is in neuroscience. There's a whole area of neuroscience that studies emotions called affective neuroscience. And an important part of affective neuroscience is the study of interoception. Interoception is our sense or our perception of the physiological condition within our body, okay? Interoception allows us to maintain homeostasis. We can allostatically adapt when our brain anticipates our body's needs and prepares to satisfy those needs before they arise. We get hungry in anticipation of meals. Interoception is increasingly recognized to impact cognitive and emotional processes. And some folks even link it to consciousness and body awareness. Like it's the key to the mind-body connection. Interoception is essential for well-being. Neuroscientist Antonio Damasio even asserted that we are not thinking machines that feel, we are feeling machines that think. We believe our powers of interoception evolved for allostasis to allow our bodies to respond to the demands of the environment. And it's actually awareness of our internal state that enhances emotional regulation. And while it might seem that really feeling your feelings, especially right now, could hamper emotion regulation, it's actually the opposite. Our ability to perceive our bodily signals through interoception facilitates the down-regulation of emotional arousal, which I find to be kind of comforting right now. And I think an important part of understanding this is that interoception relates to embodied emotion. Simply put, we feel our emotions in our body because they are physiological. So I might say that when we see this in our language, I might say, I'm full of joy, or I'm over the moon with love, right? Like we say these bodily things to describe our emotion. This idea shows up in a movie called Inside Out, and each of the characters is supposed to embody an emotion. The cute orange one in the front with all of the baggage is anxiety, which I think is pretty illustrative. So Raquel Lee last year, she is preparing to go to graduate school, did a pilot study looking at how interoception correlated with well-being. And basically there's this assumption that different people are in touch with their bodies in different ways, right? Some people are more sensitive than others. We have varying levels of interoceptive awareness and accuracy. We collected some pilot data. The N was only about 40, and we used an existing interoceptive accuracy scale. It was very much positively correlated with folks who had a lot of positive emotions. It was negatively correlated with the amount of negative emotions people have. And it was inversely correlated with dietary disinhibition. So folks who are lower on interoceptive accuracy were more likely to feel as though they were in like control of how much they were eating at times, which is pretty straightforward. And I also think it's notable that it was not related to diet quality. So it wasn't just related to any measure of eating, like there was some sensitivity in the scale. And I think it's also important to note that that earlier metacognitive data that I mentioned, in part is interoceptive in nature, because we asked the participants to look into their mind, to think about their processes, and to examine them. And that relies on some awareness of what's going on inside of our body. Okay, so now we need to put on our neuroscience glasses for a bit. If we want to study the relationship between, thank you, interoception and well-being, we need to study the insula. The insula is active when we are sensing our internal environment within our body, but like every area of the brain, it does multiple things. It's also active when we taste our beer or our tater tots or our salad. And this region is part of an important network in the brain called the salience network. So the brain here, the small brain in the middle, you can see there's sort of a shell-shaped thing there. That's the insula. So it's insulated, it's cortical, but it's sort of tucked within the cortex underneath the temporal lobe, the frontal lobe, right here. So the insula is insulated right here. On the bigger brain, it's labeled AI. Oh, I don't have my cursor up there. So the purple circle in the middle says AI and PI. It's anterior insula and posterior insula. And as part of the salience network with the anterior cingulate, which is labeled as small d ACC, okay? So the two hemispheres of the brain are largely separate, but the cortex folds down within, and the anterior cingulate is right here on the internal part of the cortex. So I'm gonna pass this model around. Two things. One, it's very expensive. UW Tacoma owns it, not me. Because it has small pieces on the middle, because it's pretty accurate for a brain model, and because it's actual weight, which I think is really cool. So I'm gonna pass it around and you can kind of peek in and see the insula. If you wanna take it all the way apart, I would just ask that you wait till that. Okay, so the salience network. There's several important networks in the brain. Salience network is one of them. Another one is the central executive, that like top-down cognitive control that makes us be responsible and make good decisions. The salience network integrates our sensory signals, like we can see here, our internal sensory signals from the body, our exteroceptive sensory signals from our five senses, as well as motor information about where our body is and how it's moving, and metabolic information. William Selly called this the salience network because what this network does is it takes these signals from our body and from the environment, and it provides some emotional information or the affective realism to make it more salient. So food isn't merely an energy source, but rather it's delicious or disgusting. It adds the emotional salience to our sensory experiences. When we attempt to regulate our emotions, often the focus is on this top-down cognitive control of negative emotions, basically talking yourself out of feeling bad, right? But what about the other side of the coin? What about encouraging yourself to feel good? Camp and colleagues tried to do just that while they were recording some functional magnetic resonance imaging data. fMRI can show us a few things, but the idea is that it shows us the areas of the brain that are using oxygen, or they're busy and they're active, okay? And what they found is that when participants were instructed to intensify the emotions you're currently experiencing while they were looking at pleasant images, like a cute kitten, it increased processing in the salience network, which is not surprising. Oh, here we can see, like over to the sides is the insula, and then the anterior cingulate, and then down here, the insula and the anterior cingulate. But it also increased processing in the ventral striatum, which contains pretty important reward centers in our brain, the very areas that are activated when we engage in other goal-directed or motivated behaviors, like eating yummy food, drinking beer, hugging someone we love, or getting a good grade. I was hoping there'd be some students here. Yeah. The ventral striatum is like kind of at the base right here. Oh, that's right. Sorry, we can't see. The ventral striatum all the way over on A. Oh, okay. So I'm going to show the insula and then the anterior cingulate as well. Okay. But you know where the insula is because you have the model of the brain. Trust me, they're lit up. Okay, so exploring the neurobiological correlates is now much easier and accessible than it was when I first started at UW-Tacoma. There's been a like prolific expansion, not just of neuroscience, but also of open neuroscience resources, some that are functional magnetic resonance imaging, and others that are connectivity atlases of the brain. So this first diagram, on the left, the top area is the anterior cingulate. Then second one over shows the insula. This is the intrinsic connectivity of the salience network. It was one of the first studies that was published on it, and it has since been replicated. However, this notion of networks that dominate our processing actually came from non-human studies. So here we can see data from the Allen Institute's Mouse Brain Connectivity Atlas. Then the Allen Institute has a mission of open science. And we can see on the furthest left is the mouse version of the anterior cingulate, and then the anterior insula. And these areas with anterograde and retrograde tracing are intrinsically connected in rodent brains as well. So this is not unique to humans. So my goal, in part, in preparing this talk, is to motivate myself to brush up on some of the technical skills that I need in order to access and work more with open neuroscience databases. Next quarter, I hope to recruit some students to work on preparing protocols. And hopefully, by looking at the brain more directly, we can move past some of the confounding factors that have complicated the study of eating to better understand well-being. Before I give this back to Jim, thank you to my boss, the School of Interdisciplinary Arts and Sciences, and my colleagues, the Undergraduate Research Committee of Practice for funding from Heather Dillon, and listing all of the students who have worked on this over time.
Great. We'll give her a short break. Anybody who has not gotten a raffle ticket from me, please raise your hand. We'll get you one of those. All right, so if you're wondering what we're giving away, so we do give away advertising. So you get Think and Drink toque, which is definitely appropriate this time of year. And as I'm learning from my students, it actually is even appropriate in the summer if you're from around here and not as old as me. So, so we're gonna read out the tickets. It's really hard to mix tickets in a beer glass. All right, so winning, what was that? Yeah, totally. Last three digits, 756. 756. All right, congratulations. All right, and we'll give away another one here. Last three digits, 776. 776. Hey, we got a winner. All right, just to let you know, so one, we still have more hats to give away at the future talks, probably through middle of 2025. So hopefully you'll come back and win one then. After that, we move on to a different type of clothing. I'm not sure what exactly yet, but we'll figure that part out. So without further ado, we'll take questions and bring up our wonderful speaker. Question.
Yes, Amber.
I was wondering if there was read about the research, if there's a difference.
Yeah, so the Salience Network.
Can you?
Oh yeah, so the question was whether or not there's a difference in like perceptual processing of a meal in neurotypical and aneurotypical folks. Yeah. The Salience Network, and as well as its like complementary networks, including the central executive, are mostly studied in reference to psychopathology. And one of the populations that there's more data and fMRI data about is like neuroatypical or like neurodivergent populations. It's expected that the balance between the central executive and the Salience Network is different in a lot of forms of psychopathology. Yeah, as well as in just other ways of processing. Yeah. Other questions? Yes.
So what does this line of thinking or way that you're designing this mean for potential sort of people that want to change how they're eating or want to think about how they're changing? So
Yeah.
what does this mean?
So I'm not a clinician and I don't make any references. I almost wore my shirt today that said riots, not diets. I think generally though, the idea is that attending to the pleasantness of experiences in some way is more powerful than trying to like change and limit oneself, right? So like diets are restrictive by nature And a lot of the idea with enhancing positive emotions is that if you attend to the minute details in an experience and what's pleasant about them, it's called the broaden and build model, then you can enhance positive emotions in that way. You just have to be careful not to go overboard. Yes, Barb. Why the asterisk? Oh, yes. So because of the amount of anti-fat bias that comes with the label of obesity and how it's linked to the BMI cutoff rather than the health outcomes, individuals who are advocates in this area have started to treat obesity like a dirty word. And so in reference to their advocacy, I tried to do that as well. Patrick. Yeah, so I have attended a few professional developments around computational work in neuroscience this quarter, and I have some more planned. I'm going to recruit some students to work with me on developing protocols, which I'm excited about. Yeah, hopefully students that are better at it than me. Yeah. Yes, Leighann.
I wondered if the factors used, the social desirability factor, that could influence how people take surveys. Is that also connected to like perceptions around anti-fat bias, or maybe even the reverse, where people want to seem like they don't have that bias, but in fact they might? I'm just wondering if you know anything about that.
Yeah. That's a big, so Leighann was asking about the social desirability bias and if it's shaped by someone who perhaps is like perpetuating that bias or experiencing that bias themselves or as a response to it. And I think it's pretty clear. You can actually measure someone's like propensity to answer in a socially desirable way. And you can see folks that score high on those measures of social desirability kind of highlight this group they live in. So if they live in a place where there's anti-fat bias, like the United States, then they answer in that way. But if they were to live in a more body inclusive society, which there's few, but in one of them, I would anticipate they would answer in a way that's reflective of the society or context in which they live. Yeah. Yes, Izzy. What motivated me to go towards like eating the newer idea or the old work? The newer work? I think that, you know, when I was in graduate school, the whole outlook of everyone that studied ingestive behavior was a focus on weight and obesity. And that influenced my research, and it influenced like almost every publication in the area of adjusted behavior for like 40 years. And it's just really limiting and oppressive. And I wanted something different. Yeah. Jim, did you have your hand up? Yeah. Yes. So there is, yeah. So the question was, is BMI related to health outcomes? And it like higher BMI is reliably correlated with some types of heart disease with type 2 diabetes. The issue is that we can measure those directly, right? So we have the technology. It is relatively affordable to measure insulin sensitivity, to measure like cardiac indicators. And so my point is, if we can do that, we should. Why should we ostracize shapes? Especially like, yeah, when we could just measure those. Yeah, it would be different if we couldn't do that. Yeah.
Questions? All right, let's thank our speaker one more time. Thank you all for coming. We got a little bit extra time, so you can hang out for a little bit. When you do leave, if you wouldn't mind actually throwing your stuff, the glasses and trays in there, and throw away the trash. It helps the crew that has to clean up after us. And it keeps them happy with me, so we can keep coming back here every month. So hopefully, stay tuned for December 10th. We'll let you know if we get a speaker lined up. So check out the website, and we'll be sending out emails. Otherwise, we'll see you in January.